Hi, Mr. YS here. Here's a tutorial on DNA structure for HL. So first thing, try and draw DNA structure. Pause the video and try and recap the whole of DNA structure. Using a pencil, try and draw and label. Okay, here we go. Here is what you should have. So we are looking at two strands that are anti-parallel, and around those strands we're going to try and label every feature of them. First of all, within these nucleotides, we're going to try and label all of the positions of the carbons. And so we can show that our strand is running 5' down there to 3' at the bottom here. So that's running 5' to 3'. That's the 5' end, so the end nearest to the number 5, carbon 5. We can also start to label our covalent bonds in between. We can label our hydrogen bonds as well. And we can show which ones are making 2 and which ones are making 3. This would be A to T, AT2, GC3. So 3 hydrogen bonds, and we would have to make sure that we also label them fully. A and T is not enough for the marks. Okay, we could then label our phosphate. This bond actually, these bonds between, these are phosphodiester. So you might see that written. You don't really need it for the marks. But these are the covalent bonds. That's the phosphate group. Then we're going to label our deoxyribose, which is our pentose sugar. We need to label our nitrogenous base. So nitrogenous base there. We can label these. So that was 5' to 3', and this one is 3', and up here that's the 5'. So these are anti-parallel strands. They're polynucleotides because we've got many nucleotides, and so when you draw this you have to draw at least 2, probably better to draw 3. Okay, what else can we label here? We could talk about which ones these are. So pure green apples, the purines are G and A, and the pyrimidines are the remaining ones, T and C. So a way to remember that is pyramids took centuries. So that's a T and that's a C. So over here we can see the difference between those. That our purine was this double ring structure, pure green apples, or the other way some people prefer is pure as gold. Purines are A and G, and then pyramids took centuries, pyrimidine and thymine and cytosine. So one other thing about DNA structure is that it's forming a helix, and this helix has a regular repeated pattern, and a bit like we saw in protein structure where we've got an alpha helix, The alpha helix is held together through hydrogen bonding. And this is what's holding together the DNA. We've got hydrogen bonds here, which are leading to this structure. A double helix held together through hydrogen bonds. All right, what is the evidence then that DNA is the molecule of inheritance and that it does pass information to the next generation? So they had two candidates. One of them was DNA and the other candidate was protein. And they picked these because they're both complex molecules. And they were thinking life is very complex and we need something that's complex to pass on information. And protein is certainly complex because we have 20 different amino acids. We only have 4 different bases. So in some ways, just looking at it, maybe you could think that protein was the better option. So what they did was they investigated, and this is the classic Hershey and Chase. So like the Hershey bar, like the chocolate. Hershey and Chase experiment. And what they did was they took something over here which infects bacteria. So this is called a bacteriophage. And what it does is it lands on bacteria and then it infects them and it reproduces in them. So here they had something perfect to try and follow the molecule of inheritance. And they're trying to follow the molecule of inheritance for this particular virus. So we can have a look at what it's made of. It had a coat made out of protein and inside was DNA. And so which is passing on the information? What we need to do is we need to distinguish between these two and we need to track them. So just stop and think about how we can track molecules that are so small that we can't see them. What technique could we use to follow them? Well, the technique used is to do with radioactivity. So we use this. Radioactive traces can be injected into the body to follow and trace blood flow. And radioactivity could be used here to find out where is the protein going and where is the DNA going. And so we need something that's in DNA but not protein, and in protein but not DNA. Stop and think about if you remember the elements that made up DNA and protein and how they might be different. What could we use to track the movement of one and not the other? So for protein, we have carbon, hydrogen, oxygen, nitrogen, and then some sulfur. So it's chons. Whereas for DNA, we have carbon, hydrogen, oxygen, nitrogen, and phosphorus. So what's the difference? Well, we don't have phosphorus in protein and we don't have sulfur in DNA. So these were two good candidates. If we can make these radioactive, then we can track the radioactive phosphorus and track the radioactive sulfur. So what they did was they grew the bacteriophage in a medium which is an environment. They grew it in a broth which is like a liquid. Okay, so in the broth, in the liquid, they put radioactive sulfur. So why is it radioactive? Well, it will emit radioactive particles. It will give out radioactive particles which can be detected. So they grew the bacteriophage where there was radioactive sulfur. And sulfur is part of proteins. And so the radioactive sulfur would go into parts of the phage, like the coat, which is made out of protein. But it wouldn't go into the DNA because the DNA doesn't contain sulfur. And then they mixed it with the bacteria that it could infect, and then the phage will do what the phage does. It will attach to the outside of the bacteria and it's going to inject its genetic information. So they allowed them to mix together, and what would be happening would be that the radioactive protein in the coat was outside, the DNA got injected in, and then they blended the mixture in order to cause kind of shaking. And what this would do is it would dislodge, it would get the virus to fall off. So they were no longer attached to the outside of the bacteria. And so now what we've got is separated the bacteria and any parts of the virus which were just outside and attached to the membrane. Because the virus attaches by binding to receptors. And so now through blending it would fall off. Then what they did was they spun it around at high speed. And this is called a centrifuge. A centrifuge spins around at high speed so that things that are heavier fall to the bottom. And so the thing that was biggest and heaviest would be the bacterial cells, the whole thing. They would fall to the bottom, whereas the lighter things, the virus particles which were left outside, the thing that wasn't passed into the bacteria, would be on top. And this part here, the liquid, is the supernatant. And this bit at the bottom is the pellet. So when they tracked it, they found in the supernatant they had radioactivity. And they had that radioactive sulfur which was in the protein coat. And down here in the bacteria, they had only the DNA and they didn't have any radioactivity. So this is showing that the radioactive protein coat was left outside and therefore it wasn't passed into the bacteria. And then the viruses that were reproducing, when they tested the viruses also did not have radioactivity, the new viruses which were being made. So if the new viruses were being made using the genetic material, then that genetic material cannot have had radioactive sulfur. And so therefore it's not protein that is being passed on and used by the viruses to make more of themselves. So now in the second experiment, they're trying to confirm that it is in fact DNA that's carrying the genetic information. So what they did was they did the same thing. They got bacteria, and this time, instead, the radioactively labeled element was phosphorus. It was 32P, and this is radioactive. And they put it into the bacteriophage. But in this case, it was only going into the DNA. So only the DNA is going to be labelled. It's not going to be the outside. And so they then mix them together and allow the bacteriophage to infect the bacteria. And what they would get then is that inside, the radioactive DNA would be injected into the cell, and then they would give it a shake and a blend. This would then dislodge the bacteriophage off the surface. And after blending, and then separating, what they would have is that the coats up here would not be labelled. There was no labelling of the coats. They were not radioactively labelled. But down into the bacteria, they then found that there was radioactivity labelled, so I'll just highlight it as yellow. The radioactivity had gone into the bacteria, and it was in the pellet at the bottom, but it was not in the supernatant. And then subsequently, any of the viruses which were going to come out of this, which were going to burst out, the viruses would be labelled. So that's showing that the radioactivity is in the DNA. The DNA was passed on from the bacteriophage, and then it was reproduced inside the bacteria, and then the next generation of viruses would also be radioactively labelled with this 32P. So here's a quick recap. So it was the Hershey and Chase experiment. They radioactively labelled 35S for sulfur, and they did that to label the phage, the bacteriophage, to label the protein coat. And they found that the bacteria infected with the phage with the radioactive protein, well, the protein was left outside. There were no radioactive phage produced, or none passing on inside into the pellet. But when they labelled the radioactive 32P, the phosphorus, because it's normally 30P, to label the DNA, because of the phosphate group, then when they infected the bacteria, the 32P was inside the phage and they could detect the radioactivity, and also in the phage produced after they contain the 32P. So therefore it's DNA that's passed on, and it is the molecule of inheritance.